And we're going to see this action potential come down and cause neurotransmitter to enter the synapse. And that neurotransmitter is going to land on receptors that are on the surface of the muscle cell in the synapse. Okay? And this is really the beginning of a muscular contraction. Okay? So this action potential, what we call an action potential, is going to propagate. Do you guys know what the word propagate means? It means to spread. Okay? So this action potential is going to propagate out from the synaptic cleft and kind of move along the surface of the muscle cell. And as it does, what we're going to see is sodium ions that have been accumulated outside the muscle cell. These sodium ions are going to actually end up entering the muscle cell down what we call T-tubules. And T-tubules are really like tubes that penetrate the surface of the muscle cell. And they do so and they connect with the sarcoplasmic reticulum. Right? Does anybody remember what the sarcoplasmic reticulum is? This is, I'm drawing it here like this, but we'll talk about what it actually is. The sarcoplasmic reticulum is essentially a giant reservoir that is going to hold calcium ions. And these sodium ions that are rushing into the muscle cell are going to flow down, and some associated proteins, and I'm not going to nail you down for those associated proteins, they're going to cause calcium ions to come rushing out of the sarcoplasmic reticulum and into the muscle fiber at large. And these calcium ions that are doing that are going to be ultimately what triggers the contraction. Okay, so if we zoomed in on the myofibrils, okay, so this is this whole thing is the muscle fiber. If we zoomed in on the myofibrils, what we would see, you guys might remember this, we have the actin, right? We have the actin filaments. And we have the thicker filaments, which we call the myosin filaments. And these thicker filaments, called the myosin filaments, are going to want to, chemically, they're going to want to bind with the actin filaments. But they really can't, because the actin filaments have these other proteins that are literally acting like a blockade. They're literally stopping the myosin from binding with the actin. They're preventing binding. So the proteins that are preventing binding, those are going to be, it's actually a complex, what we call a complex of proteins. Why is it doing that? There we go. Called troponin and tropomyosin. These two proteins together form a complex, and they block myosin from binding with the actin when no Ca, no calcium ions, are present. Okay, so now we're going to introduce calcium ions. And when we introduce calcium ions, those calcium ions come filtering in. I'll stick with the same colors here. Those calcium ions come filtering in. They're going to bind with the troponin, and they're going to cause the troponin to move the tropomyosin out of the way. It literally drags it off the sites that myosin wants to bind to. And once it's off those sites, we're going to see the myosin head can bind to the actin filament. Okay? And this myosin binding causes a structural chemical change with myosin, and myosin changes shape. So if we looked at myosin in its relaxed state, or what we would call the released state, myosin heads, they kind of look like this. Okay, so this is relaxed myosin. And myosin, after it's bound with actin, we'll say post-actin binding, it has changed shape. It has pulled the actin filament. So what we are seeing here is the actin filament being pulled in that direction. So if you look at this diagram up here, what we're going to see is the overall length of the sarcomere is going to shorten because myosin is going to pull the actin filaments in, and that's what shortens the sarcomere. Okay, so again, that's another term that you're going to be held responsible for. You need to know that this is called the sarcomere, and this is the basic unit of muscle, right? So this should be a review. Do you guys remember seeing this before? If you joined the class and you were on the class, you definitely saw this before. This isn't news, right? This is not news. This is old news. So how do we get myosin to go from this, we'll call it flexed state. How do we get it to go from flexed back to relaxed? How do we get it to go from flexed back to relaxed? You may know? Yeah, we're going to add in some ATP. So this is where ATP does its thing. So if I add in some ATP, there's my energetic ATP, boom. If I add ATP in, this releases myosin. I didn't want to type relaxed or write relaxed. I wanted to write relaxes. I don't know why I wrote relaxed. And it releases the myosin and it relaxes the muscle. Okay, so that allows the resetting of it. All right. If you're like, man, I can't remember a single thing here. I don't remember any of this. What I will tell you is this is all in one of the other videos that I'm pretty sure I posted. Plus, it's also really highlighted nicely in that sort I can't think of the movie maker for it. But that video I showed or shared with you the other day about rigor mortis. Okay, so that's a nice review of all these steps in the context of rigor mortis, which is something that happens after you're dead. And the reason why that makes a real nice illustration of that is when you die, one of the signs of cell death is calcium release from the muscle cells. The sarcoplasmic reticulum no longer is pumping calcium in, so it just kind of lets it go blah. And once it goes blah, all the myosin binds with the actin and contracts and uses up all the ATP. And once the ATP is all used up, then the muscle can't relax anymore. So we end up with a state where the myosin is constantly in a state of tension in rigor mortis because the ATP isn't present to release. Okay? So I'm going to call that the review of muscular contraction. That's pretty much all the events from the action potential through the depolarization with the sodium ions rushing in, calcium ions being released from the sarcoplasmic reticulum, causing the troponin tropomyosin complex to move off the actin, myosin binding to the actin, myosin bending and contracting, shortening the sarcomere, shortening the overall length of the muscle, and then ATP being used to release and relax the muscle, or release and relax the myosin to allow the muscle to release and relax. So that's the whole thing of contraction in a very brief summary. So does anybody have any questions